As we consider this subject of enzyme behavior, let's look more carefully at the nature of the active site, where enzymes exercise their incredible catalytic muscles. We might ask some relevant questions about the nature of the active site and how that relates to enzyme activity. Which amino acid residues occur in the active site and how do they participate in catalysis? Is the relative position of these residues also important for catalysis? How does it actually work? Let's return to a familiar friend, chymotrypsin. We know what it does in terms of the hydrolysis of peptide bonds. We now want to consider how it accomplishes this. We've already seen that it can use the artificial substrate paranitrophenyl acetate to produce paranitrophenolate and acetate. As this enzyme was studied, it was found to employ a two-step mechanism. In step 1, the enzyme breaks the bond and releases the first product. In the process, the acetyl group becomes covalently attached to the enzyme. The second step involves breaking that attachment to release the second product, acetate, and return the enzyme to its original form. If we follow the release of the two substrates over time, an interesting pattern appears. As the red curve below shows, there is an initial burst of activity, demonstrated by a steep slope to the curve. The release of the paranitrophenolate molecules occurs more slowly after this initial burst, a shallower slope. Now notice the blue dashed line, showing how this correlates to the release of the final product, acetate. From this we can see that the initial burst has to do with formation of the covalent bond with acetyl. Notice that no acetate is released during this phase. The second step of acetate hydrolysis is slower. In other words, the population of enzyme molecules readily binds substrate and releases the nitrophenolate product. That's our initial burst. But because hydrolysis of acetate is slow, the enzyme molecules must wait for that to occur before they can bind another molecule of substrate and release more nitrophenolate. Chymotrypsin is an example of a class of enzymes known as serine proteases, which use an active site serine residue in the catalytic hydrolysis of peptide bonds. Studies were conducted to prove the presence of a particular serine residue in the active site and the key role it plays in the mechanism. Diisopropyl phosphofluoridate, or DIPF, was reacted with the enzyme, resulting in covalent attachment of a portion of the reagent to the active site serine. This covalent attachment rendered the enzyme inactive, since the oxygen atom must be unattached and available for nucleophilic attack. Another important residue in the active site is histidine 57. This was also confirmed by covalent attachment of a chemical group from TPCK ketone. This attachment also resulted in a loss of activity, thereby proving its essential role in the catalytic mechanism. Aspartate 102 was the last of the residues proven to be critical for catalytic function. X-ray crystallography revealed that these three residues form a spatial unit within the active site, as shown below. Note that though they are far apart in primary structure, they have been brought into close proximity due to the tertiary fold of the enzyme. These active site residues are referred to as the catalytic triad, composed of serine, histidine, and aspartate, each with their part to play. Clearly, the architecture and composition of the active site has everything to do with how an enzyme functions.